Good morning, everybody. Let's get the chat up. All right, just wait another minute or so for people to roll in, and then we'll get going on prokaryote DNA replication. Okay, so if I remember correctly, we ended up around here. I'm sorry, I'm struggling to see kind of the old videos. I need to fix my computer. Uh, where did we end up at the end of the lecture on Wednesday? Did we cover Okazaki fragments? Anybody? No? All right. Okay. So hopefully I'm not just speaking to myself. Um, okay, just as a refresher, wouldn't hurt even if we did cover it on Wednesday. Um, the anti-parallel double helix deal, the fact that you have DNA going in different directions means that, or the two opposite directions, means that you're going to have to synthesize the one of the strands backwards, right? By flipping the strand over and synthesizing in short stretches. So even though the direction of replication is that away, opposite direction to that of the replication fork, which is going that way, the actual movement of replication is from right to left, right? So you synthesize this fragment, and then you release the strand, gather it in again, you synthesize this fragment, and then you synthesize this fragment, right? So you're kind of doing it backwards in little chunks, little steps, right? And that generates these things called Okazaki fragments, which vary in size depending on the organism from 1 to 2,000 base pairs in bacteria to really short ones, actually 100 to 200 base pairs in eukaryotes. Now, if you think how big a eukaryotic chromosome can be, we're talking in the order of potentially 100 million base pairs. That's an awful lot of Okazaki fragments, right? That's why this is called the lagging strand. Because if you think about DNA replication as a whole, going from one copy, one chromosome essentially to two, right? For one of those, it's going to take a lot longer to put all of these bits together, right? Than it will the leading strand, which is made of much larger pieces. And essentially, this is what it looks like in the different organisms. So eukaryotes, bidirectional, you're going to have a leading and lagging strand per replication fork. Same with bidirectional theta replication. The only one that doesn't suffer from this is rolling circle replication, which only has a leading strand. Right, that is it. Okay, so what we're going to do now is we're going to start on DNA replication in prokaryotes. And part of the reason is that prokaryotes, generally speaking, are a little simpler. They still do the same things, but they do it with less of an array of actors, essentially, right? So it's a little more straightforward to learn prokaryotic biology than it is eukaryotic. So that's why we do this one first. We'll be doing the same when it comes to control of gene expression later on in the semester. Okay, so again, want to get across this, sorry, my arm is kind of dead. This concept of a process, right? through which DNA replication proceeds, right? So we have to start it, do it, and then finish it off, right? And this can happen at the same time, right? Particularly in eukaryotes, but so it doesn't have to be like a linear thing, right? You have to initiate obviously first, and then you have to elongate. But while you're elongating, right, while you're replicating DNA, you can be finishing up the stuff that you've already done, right? It doesn't have to be sequential. Also want to again introduce you to a new word which we'll be using later. Sorry, I got my shot yesterday, and it means I can't move my left arm, which is kind of annoying. I have to kind of walk it along with my fingers. And that's our new word, which is holoenzyme. And this is I don't know, best way of describing it, group of enzymes, proteins that work together as a complex. Right, and so you'll come across this a lot. Really, the whole of DNA replication is, at least the elongation is done with a holoenzyme, right? So these proteins are not acting independently, even though we show in many cases that they are. Like our complete understanding of them is that they act as a group, right? And this is why it's actually kind of shown with them separately, but DNA polymerases are tied together, right? So that they proceed at the same rate, kind of up the strand with the replication fork moving. Okay, so in terms of initiation, right, we have again that origin of replication or replication origin. In E. coli, it's called an ORI-C. I don't know where the C comes from, but ORI is fairly obvious. And this is recognized, this is a stretch of DNA which is recognized both by its shape and its sequence by initiator proteins, right? So as soon as those initiator proteins are produced, presumably in the early stages of cell division, they then are able to recognize this ORI-C and they wrap the DNA around themselves. And the reason why they do that is that they physically bend the DNA, they change its conformation. And in doing so, it's almost like kind of grabbing a rope and twisting it, right? Or garden hose, kind of rolling it onto the carrier the wrong way. It will pop the two strands open, right? And this is a stretch of AT-rich DNA. So it's going to have fewer hydrogen bonds holding it together. And so once that's done, this is really just a conformational change, right? So it's really just twisting the DNA in a way that forces a weaker part of it to pop open. Once it's open, however, this can easily go back together, right? There's not really anything stopping it other than this shape change that what are those dogs doing? That forces this to happen. And so next comes what are called single-stranded binding proteins, which you probably guessed it, bind to the single-stranded DNA, right? This is what we need for DNA replication, right? You can't copy something if you can't see it. And so these hold the two strands apart, right? They stop it from just kind of going back together again. And they hold it apart long enough for the DNA replication machinery to get in there, right? They don't actually play any other role in DNA replication than that, right? It's really just to stop the DNA from sticking back together. And then we have, for each replication fork, right? So this is presumably bidirectional replication because we have two copies of this, is a protein called helicase. And its job is really to kind of act as a plow, right? It's going to go, or a zipper, an unzipper, really, right? It's going to force itself down the double-stranded DNA, and in the process, it's going to push those individual strands apart. right? And so it's going to progress along the DNA ahead of the DNA polymerase, which I haven't got to yet. I don't know what they're doing. Mischief, probably. I've got 2 new fostered dogs, 3-month-old puppies, and they're full of mischief. And this is going to kind of continue this process, right? So it started here by popping open the double strands, and now the helicase is going to continue that process all the way around, literally until they meet on the other side of the chromosome. Okay, now when that happens, because you're unwinding something, if you have a fixed thing, it's like, again, garden hose, and you're unwinding it at one point, to be able to unwind it at one point, you coil it up more in another point, further down, right? And, oh, I remember where we finished. It was about here last time, right? Anyway, you get a little bit of a recap. And so if this continues for long enough, what will happen is that that uncoiling will introduce so much coiling elsewhere in the bacterial genome that it will become what's called supercoiled. It's basically like, just a tangled mess, like Christmas tree lights or those lights that you hang outside with the LEDs and stuff. I spent hours untangling one of those ones. And so to prevent that from happening, we have a enzyme called a DNA gyrase, which is a class of proteins called topoisomerases. Topo meaning shape, like topography, and isomerase meaning other, right? And so the idea is that it's going to change the shape to another conformation, right? And these can act either by cutting one strand or both strands. And so what they do is, imagine you have like this kind of like twisted up ahead of it, right? DNA gyrase will cut one of these strands, and that tension literally unwinds itself. And then the DNA gyrase sticks that strand back together again. Sometimes it's both strands get cut. It doesn't actually make any difference in terms of function, in which case it would be simply like that, right? So imagine like this, my arm is what's twisted up. Now it's just going to kind of flunk. It's like if you have a really tangled garden hose, cut it and it will unwind itself, and then you just stick it back together again. Obviously, you wouldn't want to do that every time you wanted to water the plants, because that would be really tedious. But that's the kind of idea, right? And we have these in eukaryotes too. They're typically called topoisomerases, just as a name, right? DNA gyrase is a topoisomerase. It's just, this is what it's called in bacteria. Their mechanism is largely the same. Okay, so that's dealing with the replication movement, right? That's dealing fork movement. That's dealing with opening DNA, right? So essentially, you're going to have helicase here, right at the fork. We deal with the unwinding, right? Dealing with the supercoils. That's the role of DNA gyrase. Now we need to initiate replication. And the big problem here is that DNA polymerase needs a 3' OH to add a new nucleotide too, right? So this will be the template strand. This will be the template strand, right? Where on here is a free 3' OH, right? There isn't one, right? Because this is single-stranded DNA, right? For there to be a 3' OH, it would need to be double-stranded, which is not, because you don't want it to be, right? So we have this kind of conundrum that needs to be solved, in that DNA polymerase cannot synthesize or cannot add DNA bases, nucleotides, to a template strand on its own. It needs something to prime the process. And that something is an RNA primer, right? And so RNA polymerase has no issues whatsoever sticking down RNA nucleotides on a DNA template. And if you think about it, that's exactly what RNA polymerase in transcription does, right? To generate mRNAs, uses a DNA template to synthesize an mRNA copy. right? We're not going to go quite to that extreme, right? This is really just a short little stretch, 15, 20 nucleotides long, right? Really not very much. Just enough to make sure it sticks and provides a 3' OH for the DNA polymerase to come in and add a base 2, right? This enzyme that does this is called primase, right? Because it's a primer that synthesizes primase, right? Fairly easy to remember. And as soon as that first DNA nucleotide is added by DNA polymerase, its role is done, right? It is not important anymore. It's simply to kickstart the reaction. And then once that happens, if this is the leading strand, for example, this goes on like until it finishes, essentially. How about for the lagging strand? How often would you need to add a primer for the lagging strand? Right, so let's go. Oh, where's a good picture of that? Let's go back here. For example, questions to the class. I know it's Friday, but I've got to ask at least one class, right? How often would you need to add an RNA primer for the lagging strand? You only need to do it once for the leading strand, right? How often would you need to do it for the lagging strand? I can pick someone. Who shall I pick? Ooh. Should I pick Star? Should I pick Jimena? Oh, maybe JM. JM won't mind me picking on him, would he? Would you, JM? All right. Well, okay. We're not going to sit here like staring at the screen forever. Good idea, Jelani. That's for the leading strand, right? So up here, you're going to have to add a primer here, right, to kick this off. The answer is every single time you make one of these fragments, right? So every single Okazaki fragment has, it's not shown here, but it has a RNA primer to start it, because you start this whole thing over and over and over and over again, right? And that's what causes the delay. One of the reasons what causes a delay in finishing this process, right? Because you have to get rid of those, which we'll get to in a second, right? So here, for example, you can see, right, every time you have a lagging strand, you have to have a primer, right? And you can see, I'm not sure what that blue thing is. I think that's primase, maybe. Never fully explained, to be honest. Okay. I've already done this. Okay, so in terms of elongation, right? We have a whole range of different DNA polymerases in both prokaryotes and eukaryotes, right? We'll get to the eukaryotes, when will that be? Probably on Monday, I'd guess, right? Or maybe today if we have time, right? So and they do different things, and these are very kind of specialized proteins, right? They have a very particular job to do. It's a little simpler in E. coli because we basically have one, which is annoyingly called DNA polymerase 3, right? This is the one that does all of the synthesis, essentially, right? So it does the bulk of the DNA replication in bacteria. We have another one, this is actually called one, right? This is needed for what we'll talk about in a little bit, essentially fixing those primers. And the other ones are involved in DNA repair, right? So essentially, once a bit of DNA is removed, right, to fix an error, it restarts replication after that. Or if there's a piece of damaged DNA, it will allow DNA replication to continue. Quite often, these things only work for a short stretch, and then they fall off, and DNA polymerase 3 comes back in, and continues the job. Okay, so this is really just elongation, right? This is just going to keep happening. For leading strand, it continues essentially until it stops. Lagging strand is constantly done in these short stretches with a primer and a little bit of DNA and then another primer and a little bit of DNA. Once that's gone on far enough, right, now it's kind of moved off into the distance, right, essentially, like crew line, train track. Then you have to come along and finish up the process, right? And that largely involves stitching together the lagging strand, right? So we have some problems. One, we have RNA in our genome, right? Or the bacteria does. That you do not want, right? Because of a whole bunch of reasons, right? You wouldn't be able to copy that RNA again with DNA polymerase because it just wouldn't work. It'd be kind of easy to degrade. It wouldn't be very robust. So it'd be a poor storage medium for information and other reasons, right? So one, you don't want RNA in your genome. Two, those fragments are not stitched together, right? So in these lagging strands, right, these Okazaki fragments have a gap, right, in the backbone. So not in terms of whether or not bases are present, right? Because every single base on the template strand, the gray strand here, has a complementary base kind of next to it. But it's in the backbone, right? So there's if you look ever so closely, better eyesight than I have, right? You'll see there's a little 3' there, right? So there's an OH there at the bottom of the last sugar, and there's a phosphate group, 5' here, attached to the first sugar of that primer, right? So this backbone is not joined together. If you melt this DNA, if you heat it up to 100 degrees or so, what will happen is that you'll get one large piece of DNA, right, which is the leading strand, and lots of small pieces, which are the Okazaki fragments. And so you don't want to have your genome, or at least one half of it, right, one strand, as fragments, because as soon as you need to open it up to do anything with it, to copy it again, to transcribe it, to express genes, it will fall to pieces, right? So those are the two problems that you need to fix. And that's what happens at the end, or really, truthfully, as DNA replication proceeds further down the genome, you start fixing these problems, right? So we need to get rid of the RNA primer, one thing. We need to replace those RNA bases with DNA, and we need to basically seal the backbone, right? Join those sugar and phosphate groups together so that when they come apart in the future, it's just one continuous piece of DNA. All right, any questions so far? Okay, so the bulk of this work is done by DNA polymerase 1, which we mentioned further up. And it has two functions. One, it chews up the RNA primer. It literally breaks the phosphodiester bond between the RNA nucleotides and ejects them from the template strand, right? Which is what you see here. This being the template strand at the top. These are the RNA primers. And then once it's kicked an RNA nucleotide out, it then puts in a DNA nucleotide and joins them together, right? So it creates that phosphodiester bond. And it does that until it hits the beginning of the next Okazaki fragment, the next chunk. This does happen for the leading strands too, but it only needs to happen, twice, essentially. happens over and over and over again for the Okazaki fragments, which is why we talk about it in that context. Okay, so it's both a nuclease, right? It destroys DNA, or actually destroys RNA in this case. And it's also polymerase in that it creates DNA in that space, right? Because you don't want to have a gap. It's a major problem in lots of different ways. So once that DNA polymerase 1 has done its job, it leaves a gap in the backbone, right? Such that there's this base here, denoted by the 3, is not, bonded to the base denoted by 5, right? The 5' end of that fragment. And so what we need now is another enzyme. This is called DNA ligase. And this uses ATP as energy to create that phosphodiester bond, right? And so just to, is it in here? Is this a good one? All right, this is essentially the phosphodiester bond that we're talking about here, right? The line joining the sugar to the phosphate. I think there's another picture further up. Where are we at? Here we go. Right. This is a phosphodiester bond that DNA polymerase and DNA ligase is creating here between the O on the, or the OH really, on the 5-carbon sugar and the P of the phosphate group. And really, once that's done, for that chunk of DNA, DNA replication is largely finished. Not quite, nearly. But the DNA fragment is, the new strand is whole, right? That's the main point. And then it goes on to the next primer, and the next one, and the next one, right? So we have to do this for every single RNA primer in the lagging strand, which, as you can imagine, takes a while. Okay, so typically termination ends when the replication forks or bubbles meet. Sometimes we actually have what are called termination proteins, which recognize specific sequences and stop things from proceeding any further. That's quite a little bit rarer, to be honest. However, right, we haven't finished because, I told you how good DNA polymerase was at making sure the right bases go together. Well, it is pretty good, right? It tends to make, I don't know, one error in around 10,000 bases, give or take. But sometimes, for reasons which aren't fully understood, just random chance, I guess, the wrong base gets in, right? So we end up with, say, a T being bound to the guanine instead of a C. Ooh. And so we need to fix that. And we need to fix that real fast, because if we don't, then the next time that DNA gets replicated, that change will be incorporated. And so that mutation becomes fixed. And then depending on what that mutation does, that might be a bad thing or not a bad thing. It's rarely a good thing, but typically it's a bad thing, or it just doesn't have any effect. But either way, you don't want those things to accumulate. Now, for bacteria, an error rate of 1 in 10,000 isn't terrible. That would be, well, like 10 to 20 mutations every round of cell division. But if you think about how many times bacteria divide, even that number will become an issue really, really fast, right? If a bacterium can divide every 20 to 30 minutes, it's going to do a lot of divisions. Obviously, in larger organisms like eukaryotes, it's an even bigger problem, because one error in every 10,000 when you have 3 billion bases is really not a good thing, right? And so all DNA polymerases, the main ones, have what's called a proofreading mechanism, right? And essentially that's like how when you write something, or I write something, you see a spelling mistake, or even now we have Word, right? It will do it for us. As soon as you make a spelling mistake, you go back, get the wrong letter out, put the right letter in, and then continue. That's essentially what proofreading is in DNA replication, right? So all of these DNA polymerases have what's called an exonuclease activity, right? Exo means from the outside or the end, endo from the middle or the inside, right? And so these polymerases are able to, if they stop and recognize it, they can cut off the wrong base, right? That exonuclease activity. They can basically go and they go, ooh, don't like that. And they kind of go back a little bit, snip it off, and then put a new base in, right? And so this only works if that error is detected immediately, right? If the DNA polymerase moves on and adds another base here, right? It might be the right one, say a C. What are we at? No, a G. If we add a G here, that proofreading is not going to work anymore. Now you have to rely on mismatch repair, which occurs later, right? Oh, there's my wife back. Okay, so essentially the idea is that the DNA polymerase recognizes that that's wrong. Don't know why I put it in anyway. I don't think they actually do these things on purpose. It will then cut that phosphodiester bond, right, that it just made, and it will kick that nucleotide out so that the correct one can come in, a thymine in this case, can come in, it will recreate that phosphodiester bond and continue the process. Right, so this reduces the number of errors by quite a lot. I think it's about another about 1 to factor of 100, give or take. Right, so it goes from about an error rate of around 1 per 10,000 nucleotides, and now it goes to an error rate of about 1 every 10 million nucleotides. Right, and so for prokaryotes, that's pretty fine and dandy, right? Because their genomes are generally smaller than that. But we can go further and catch any mistakes that DNA polymerase didn't catch in time, right? And this is a process called mismatch repair. And it really works on the basis of incorrect bases forming the wrong shape, right? And so if you have two purines together, right? If you have a guanine and an adenine together, for example, now you're going to have 4 rings, right? Now, oh, now you're going to have a bubble, right? If you have 2 pyrimidines, which are only 1 ring, now it's going to kind of, the backbone is going to sit closer together. And even if you have a 2 and a 1 together, like so, that are wrong, like say an adenine and cytosine, they're still not really going to fit properly. Let's make sure I'm in the right PowerPoint. Right, and so this is actually, this is a very interesting part of this, is that this has to happen very fast after DNA replication has kind of gone through, right? So again, the proofreading mechanism missed it for whatever reason, right? And we haven't yet finished the whole of DNA replication. The reason why that matters is that DNA has marks. These are called methyl marks or DNA methylation, right? And essentially what it is typically on Cs, you have a little methyl group added, right? It's a DNA mark, epigenetic mark. And you don't get that immediately after DNA replication, it takes time for the enzymes come along and kind of copy those marks, right? And so the only way that you can recognize which is the old strand, the correct one, which is the new one, the incorrect one with the error, or the daughter strand, is if that methylation hasn't happened yet, right? And so the enzymes then recognize the daughter strand as the one that is not methylated or is undermethylated, right? Because otherwise you'd end up taking out the bit of DNA from the parental strand, right? The template, thinking that that was the error, right? There's no other way of telling which is the new one once methylation has proceeded. And so when this mismatch is detected, right, so you can see here it's a kind of like a topological change, right, in the shape of the DNA. Essentially, you have another whole process of enzymes that come in, right? One is to recognize that mismatch, right? The next one is to basically go a little bit upstream, right? 5' to the mutation, which is here, and cut the backbone of the daughter strand. Then an exonuclease comes in, right, which couldn't originally chew it up because there was no end, right? But that nick creates a free end. And now the nuclease comes in, and it only works for a short period, but it's long enough to remove that incorrect base. It removes all of these ones as well, right? But it goes far enough to remove the incorrect base. That leaves essentially the same thing that we had with the beginning of DNA replication or the removal of the RNA primer. We now have single-stranded DNA. And so, as you can imagine, DNA polymerase comes back in, starts with the 3' OH on this base here, which was the one that was left behind, and then goes back through and fills in the gaps, right? Fills in those bases. And then we have a methylase come in as after this process. Actually, methylase A, that's kind of interesting. But anyway, A's and C's, I guess, are the main ones. And methylates the daughter strand. And so now this is finished, right? And so presumably further along, we're going to want to catch those mismatches before the methylase comes along and methylates that part of the daughter strand. Anybody have any questions? All right, so again, the two main things to remember here is one, mismatch equals wrong shape, right? The DNA will not look correct. It will be physically different. And two, the daughter strand has to be unmethylated or undermethylated for this mismatch repair to work. If this isn't caught in time and DNA methylase goes through, or DAM methylase and methylates the daughter strand, that's it. You're done, right? You can't go back and fix that error. That error is now incorporated into the genome. It is essentially a novel mutation, right? And so you have to be quick to be able to catch these. Otherwise, how can you tell which is the correct strand and which is the incorrect strand, right? What do you have to compare it to? Okay, so just as a kind of general summary, right? General base pairing, right, gets you in the order of one error in about 10,000, give or take. Oh no, that's 100,000. I keep saying 10,000. You know, so can't use my arm to get my phone out of my pocket. Spam call. Probably someone asked me if I want a new car warranty or something. Yeah, so that's up to about, actually is between 1 in 10,000, 1 in 100,000. It kind of varies depending on the organism. Then proofreading, actually built into the DNA polymerase itself, catches another kind of 100-fold improvement in the error rate. And then mismatch repair is another kind of gets you another like not threefold, 10 to the 3 essentially improvement in accuracy. And all told, that's between 1 in a billion and 1 in 10 billion bases per every error, right? And so that is extraordinarily low, right? So you can see how essentially, again, we have another process going on, right? We've got the process of DNA replication. Now we have the process of ensuring replication fidelity, right? The proofreading and the enzyme itself, the basic nature of DNA, right? And also the mismatch repair right at the very end. Okay, so essentially to summarize, EV. So just to summarize, we open the helix, right, with the initiator proteins. We hold the helix open with single-stranded binding proteins. We force it further open with DNA helicase, which induces an opposite coiling problem ahead of the replication fork, right? DNA gyrase runs just ahead of the helicase, and its job is to relieve those supercoils, right? That stress of the unwinding that helicase produces. And then we have RNA primase, synthesizes the RNA primers. We have DNA polymerases, one for each strand of the DNA, so we've got 2 in total. And then we have another DNA polymerase 1, removes those primers, and DNA ligase reseals the phosphodiester backbone, right? So this is like the whole process. And by and large, it's the same in eukaryotes as well. There are extra wrinkles that we deal with in eukaryotes that we don't in prokaryotes, which I'll get to in a little bit. And obviously as well, that's not in this image, we have mismatch repair proteins and methylases, right? Essentially, they're going to be kind of off the page down the line, right? Those come after these enzymes have done their job. And this goes all the way along, essentially until it meets the replication fork on the other side or what it's already done. So there's a lot of stuff going on, but if you think about what needs to happen at each step in the process, it's fairly straightforward to remember what each one does, right? Does anybody have questions? Questions. Right, so it's hopefully not too difficult to learn. It's just stuff and processes, essentially. Okay. All right, so we can move on to eukaryotes because we have a few minutes left today, and essentially today is mostly prokaryotes, and Monday is put aside for eukaryotes. We might not actually need all of that, so we may end up with a kind of a catch-up or a recap session on Wednesday, which would be quite nice, to be honest. Don't always have time for those, but sometimes we make good progress and hit that stuff. Okay, so by and large, DNA replication in eukaryotes is very much the same as replication in prokaryotes. However, there are a few things which are different. One, we have multiple origins of replication there, right? So that's just because these chromosomes are huge, right? If you started only at one point and just went, it'd be like that kind of image of a person running across a very, very flat horizon, right? It just takes forever to do it. And so we can't wait that long, right? We need to be able to, our cells need to divide more quickly than that. And even then it takes a while, right? So we have multiple origins of replications in eukaryotes, right? Up thousands potentially per chromosome. The other thing is our chromosomes are linear, right? They're not circular. The only other things that have linear chromosomes are some DNA viruses, for example, right? And that creates its own kind of problems as well, which we'll get to in the end, which is sort of tied into RNA primers, right, at the ends, and also a bigger picture of senescence. Basically, senescence is a fancy word for getting old, right? And so this linear chromosome issue creates other problems kind of down the line, which essentially limit how many times a cell can divide, right, before it's forced into senescence. And again, that's also something that gets that is escaped by cancer cells. So it also has some, implications for cancer biology. And the other thing is that DNA, eukaryote DNA, is it's actually called chromatin, right? So chromatin, let me just and chromatin equals DNA and protein. I can see what I'm writing. These are called histones. And so that that protein needs to be removed and then replaced after DNA replication has happened. And there are essentially the same kind of proteins. They're sometimes called different names. Certainly DNA polymerases have their own kind of nomenclature in eukaryotes, which is different from that in prokaryotes. And they might be slightly different in structure, certainly probably going to be more of them. And they also have sometimes slightly different roles, right? One example is DNA polymerases in eukaryotes. There's one which begins DNA replication and then falls off and another one continues it, rather than just being the one, which is DNA polymerase 3, which does the whole job. Okay, so essentially what we mean by chromatin is it's based on essentially the packaging of DNA, right? So this is how we get to fit so much DNA into a relatively small organelle, the nucleus in the cell, right? And yet still have access to it when we need to for transcription. And so DNA, rather than being allowed to float kind of freely in a mass like it is in bacteria, is actively wrapped around bobbins, essentially, right? Little balls of protein to organize the DNA. These are called histones, and there's a bunch of different histones. There's usually 4 pairs of different histones here to make up a nucleosome of 8. Oh, there we go. So it's the 2s and the 4s and 3s. And there's also another histone called because it's kind of called the linker histone, which is histone H1. And that kind of holds everything together and also creates this space between the nucleosomes. And so this is one nucleosome, which is DNA and protein. This is another nucleosome. and you have a bit of DNA in between, which basically allows those things to kind of flex, and it also gives space for these histones to kind of move up and down the DNA like that. Ha, didn't mean to do that. There you go, put that back to where it was. Okay, so we need to get these off. And then once DNA replication has occurred, we need to synthesize more of them, right? Because now we're going to have two chromosomes or chromatids, which need to be wrapped around these proteins rather than just the one that we started with before we started copying it. Okay, I think that's probably enough to get going with. On Monday, we will go through the different polymerases and also some of the other differences, essentially, between prokaryotic and eukaryotic DNA replication. All right, that's it from me. Does anybody have any questions? All right, cool beans. Thanks very much, everybody, for showing up, and have a lovely weekend. I shall see you on Monday.